All right, last thing. The last thing that you need to know is meiosis. So that's a blurry picture. Let's talk about meiosis. What is meiosis? Well, what was mitosis? Mitosis, you started with what? Somatic cell, which is anything but your sex cells. Skin cells, cells inside of your digestive system, all of that. Anything that's going to divide. What do you start with? Diploid. What happens? One division, and then you end up with two diploid. What is meiosis? What are we talking about? We're talking about making a sperm and an egg. That's a special process. Oh man, someone's getting in trouble. Somebody's got a bad attitude. They are in trouble. It's not good. I gotta make sure like I go clean something because I might get yelled at too. Um, Meiosis is making sex cells, sperm and an egg. Now, I don't know if y'all know how babies are made. Hopefully, I'm not. This is a spoiler alert here. Is that we have an egg and a sperm, and they come together when a mommy and daddy love each other, and you get a baby. You get a zygote. Now, let's imagine here for a second that this egg has 46 chromosomes and this sperm has 46 chromosomes. That's going to be 92 chromosomes. Is that going to be compatible with life here? No, it's not. Trisomy, was it trisomy 21? One extra chromosome is Down syndrome. And having like little bits of extra chromosome are generally there are some things that are associated, genetic disorders that are associated with it. So we want to try to have 46. So what do we have to do to the sperm and the egg to end up with 46? We got to get to 23. Oh, sorry. Erase the wrong one. We got to get 23 in the egg, and we got to get to 23 in the sperm. The problem is that when we do spermatogenesis and oogenesis, they come from a spermatogonium or an oogonium that have 46 chromosomes in them. So we got to go from 46 to 23 somehow. The process of that is called meiosis. So let's compare and contrast mitosis and meiosis. Meiosis, we are going to start with a diploid cell. So we're going to start with 46. But what kind of cells are we dealing with? Gametes, sex cells, sperm and an egg. Not somatic. This is going to do what? You're going to start with diploid and you're going to go through two divisions. What are you going to end up with? 4 haploid cells. The good news is this is relatively similar to mitosis. Now, this is where the definitions become important between homologous chromosome, sister chromatid. You need to make sure that you got those definitions straight in your mind, else this is where it gets confusing. All right, somatic cell, we know 46 chromosomes, 2 pairs of 23. Yada, yada, yada. Sex cells, they're going to have 23. Sperm and an egg. Oh, this is all we got? Okay. We can do this, I think. Let's come back, and that's going to be confusing. Let me open up, let me do a new slide. Blank slide, okay. All right, so we're doing meiosis. What do we got here? We're starting with what? Let's do another 2 chromosome organism. Let's do another 2 chromosome organism. So let's do this guy. Actually, we'll do 4 with this. We're going to do 4 chromosomes here. So we got to do the. Oh, we can just do 2 again. I think we can. So we're going to have our 2 chromosome organism. Oh, I'm doing this wrong. Sorry, I've been on vacation. My brain is not working. So chromosome number 1, chromosome number 2. All right, so this is our cell. What do we need to do? We need to replicate all the DNA. So let's put the sister chromatids in. Boop, 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 boop. All right, now we put all the sister chromatids in. We've replicated the DNA. Now we're ready for meiosis. So the first step in meiosis is going to be, since there's two phases, is prophase uno. We got prophase one. Prophase one is a little different than prophase in mitosis. So what happens in prophase one is that you get what's called tetrad formation. So tetra means four. So you're going to have, let me draw this a little better. And what's going to happen is you're going to have kind of these two chromosomes, these homologous chromosomes kind of joined together. And the same thing is going to happen down here. I'm going to do a little dance. So you have tetrad formation, that's what happens. Then you have something called crossing over. Now remember, in these homologous chromosomes, this is mom's, this is dad's chromosome number 2. They both have the same instructions, the same recipes on them. They're just a little different. What happens is, in crossing over, dad's is going to share a little bit of genetic information, a little bit of his recipe with mom's, and mom's going to share a little bit of her recipes with dad's. And they're going to shuffle the deck. They're going to shuffle the ingredients. So now, that's why you don't look exactly like your parents, is because they shuffle the deck a little bit. That's what crossing over is. What is crossing over? Please listen to the words. It is the exchange of genetic material between non-sister chromatids. Why is that important? Well, if you exchange genetic material between sister chromatids, they're exact copies of each other. So you're just changing the same recipe. You don't want that. So you exchange between non-sister chromatids. Now you get a little shuffling of the deck because genetic diversity is important. If we were all exactly the same, we'd all be susceptible to the same diseases. So if one disease came in like COVID and we all had the same genetics, we would all die. The species would die off. So you want genetic diversity. It allows survival of our species. Okay, you get tetrad formation crossing over. Same thing, you lose the nuclear membrane, chromatin condenses, yada, yada, yada. Okay, cool. Now what happens is you get metaphase 1. And that's when the homologous chromosomes line up in the midline. That's metaphase 1. Now this is where the magic happens in meiosis. What happened in anaphase of mitosis? What happened? You had the sister chromatids separate. In this case, a whole homologous chromosome, the two sister chromatids, are going to go together. And these two sister chromatids are going to go together. The sister chromatids for mom's number 2 are all both going to go over here. The sister chromatids for dad's number 2, they're going to go over here. Anaphase. The homologous chromosomes separate. What happens in mitosis? The sister chromatids separate. All right, then telophase, same thing. You get a cleavage furrow. You get this here, you get this here. Now, this is actually where everything goes haploid. Now, this is where the haploid thing happens, is right here. So now you go through prophase 2, or you go through meiosis 2. Very, very, very easy. It's the same thing as mitosis, except you're using a haploid number of chromosomes. All right, so what happens in prophase 2? Nuclear membrane disappears. I mean, it's already disappeared. Chromatin condenses. What happens next? Metaphase. What happens in metaphase? They line up in the middle. I kind of already had them in the middle. I shouldn't have drawn them that way. Anaphase 2. Anaphase 2. What happens in anaphase 2? The sister chromatids separate, right? Do you want to build a snowman? They separate. And this happened with both of them at the same time. Then what happens in telophase? You get the cleavage furrow. Let me draw that a little better. Sorry, guys. It's late. Or it feels like it's 11 o'clock. I know it's not. Boop, 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 boop. And then what do? We have cytokinesis. So now you got a cell here that's going to be. And we did this twice, right? This one goes through the same thing too. So then you end up with 4 haploid cells. 4 haploid cells. So let me show you what we originally started with. What we originally started with was this. We started with a diploid number of 4, 1, 2, 3, 4. And we ended with 1, 2, 3, 4 cells. Oh, shoot. We ended with, there's 4 here, there's 2, there's 2, there's 2, there's 2. That's all it is. So just to review real quick. What are we trying to do? We have meiosis. What is meiosis? We're doing gametes. We're doing sex cells. We start with diploid. What do we end with? We do 2 divisions. We end up with 4 haploid cells. We have our 2 chromosome organism. We've replicated all the DNA. We've added in our sister chromatids. Now what's happened? We get tetroid formation with crossing over. What is crossing over? The exchange of genetic material between who? Non-sister chromatids. Metaphase 1, same thing as metaphase and mitosis. They line up in the middle. Anaphase, what happens in mitosis? Sister chromatids separate. What happens in anaphase and meiosis? The homologous chromosomes separate. Telophase, next, you get the cleavage furrow. Prophase 2, chromatins condensing. Metaphase, they line up in the middle. Anaphase 2, sister chromatids separate. Telophase, cleavage furrow. Cytokinesis, 4 haploid cells.